I'm going to talk about the quest for a malaria vaccine and really try and bring together what we do in the labs at Burnet. But first, I'm going to contextualize why we're doing it and set the stage for the need for a vaccine. And I will add that I don't get a lot of opportunities to wear my vaccine earrings, so I brought them out tonight, even though they don't match my outfit, but you have to make some sacrifices for the talk. Anyway, so just to start, I will talk about the malaria vaccine in general first, and then more about the work at the Burnet. Before I jump into it, I think it's worth just looking again at the numbers. And I'm going to actually start with some good news. So the good news is that in the last 15 years, we've actually done a lot to decrease the burden of malaria. A 37% decrease in cases and a 60% decrease in deaths is a lot. And we have to really applaud the global health community for that. The bad news is that in 2015, we're still looking at a really, really high number of cases and deaths. And for every one of those cases, that's someone feeling really horrible. Goes without saying that having a disease is terrible, but then you also have to think about the economic problems that go with that. That's a child not in school, that's its parents taking time off work, and if you are earning a daily wage, that's a big brunt to your family. So there's so many issues with that. And you can see here, this is a map where everywhere in blue are countries that still have malaria. And everywhere in green are countries that have actually eliminated malaria in the last 15 years. So it is possible, it is definitely possible, and we can keep working towards this. How do we do this? So this is a picture I borrowed from The Economist magazine because they do a much better job than most of us scientists at fun pictures. And it really highlights that we're throwing everything we have at the malaria parasite. We've got a toolbox, as Professor Brendan alluded to, and in this toolbox we've got a few different things that here you can see cartoonishly shown. So the first thing we have is vector control. And I'll tell you more why we target the mosquito. They always get a bad rep because they carry the parasite. And one of the best public health measures we have is actually targeting the mosquito. And we call that vector control. That's insecticide bed nets, repellents, all sorts of things related to that. The next thing we have somewhat quick and sensitive diagnostics. We do have a lot of good diagnostics, and one example of that is actually on your table at the moment, and Lisa, who will speak after me, will talk to you more about them. At the same time, there's always a need to get more and more sensitive, but I won't go into that today. A third thing that, again, we have at the moment are effective and safe anti-malarials, but there's also emerging resistance that accompanies these. And so we don't want to get too confident yet because we still need new drugs to take the place of the old ones. So we have a few things in the toolbox, but one of the other things that would definitely make a difference is a malaria vaccine. And all of these have to work together to really hit those elimination goals. So before I go forward, let me convince you why I think vaccines should definitely be in that toolbox. So firstly, I'm sure you all know this, but a vaccine prepares our immune system for what the disease looks like without the symptoms, so that when the disease does come along, our immune system is ready to fight it, and you don't have to actually get all the horrible symptoms. So that's the first reason. And vaccines are actually one of the greatest public health tools we have at our availability. Not necessarily yet for malaria, but for many other diseases. And the evidence for that really comes from history. So vaccines have actually been really critical for the elimination and eradication of many other diseases. Professor Brendan already alluded to smallpox. We no longer have smallpox because of excellent vaccines and vaccination campaigns. And even now, we're actually living in a very historical time. We are so close to eradicating polio. We're at 99%. And obviously the 1% is always going to be hard, but we are so close because of a good vaccine. So historically, there's a lot of evidence to push for a vaccine. But everyone here has probably had a vaccine. You know they're safe, they're cost effective, because you only really have to give 1 or 2 or 3 doses, and you can last you for years and years. And therefore, they are long term. So as I said, a vaccine would definitely be great in that toolbox to work with all the other tools we have, because we just have to throw everything we've got at this Scrooge. So where do we want to go with the vaccine? So everyone is interested in developing a malaria vaccine, but it's really important that we all choose a specific goal to work towards. So organizations like the World Health Organization bring together groups of experts and they prepare goals for us to work together as a community. And the malaria vaccine roadmap actually decided that a good goal for us to focus on is by the year 2030, let's have a vaccine that's 75% effective. And you might say, that's not great, but trust me, that's a hard enough goal for us to work towards. And let me tell you why. Where are we now? There is currently a malaria vaccine. You would have read many articles about it. It's been all over the news for the last few years. This vaccine, RTSS, or I can never say it, Mosquirix, was developed by GSK, GlaxoSmithKline, and with a collaboration from many scientists around the world, but a lot of help from the Bill and Melinda Gates Foundation. So this vaccine exists, and it was the first vaccine to make it to phase 3 clinical trials. These are the really large-scale, thousands and thousands of children and infants trials. It was tested in 7 sites across 11 countries. Big, exciting, everyone fingers crossed, and then the results came out and they had some efficacy, but 26% for infants and 36% for young children. Again, some efficacy, great, but really not what we need. I think you can agree with me that we really need to set the bar higher. This vaccine now exists and it will be deployed as a pilot program in some countries, but there really is a need for all of us in the global malaria community to really keep working to develop highly effective vaccines. Let's at least get the 75% if everyone agrees with that part. So this is where we are now, but what are we doing at the Burnet? So I put up a picture of some highly photogenic scientists that I work with. We always look this good when we're doing experiments. So I just put up a picture of our colleagues, and now I'll tell you a bit more about what we do right here at the Burnet. And to be honest, I really struggled trying to decide which of the many things to talk about. So I've sort of chosen 2 things that I like, but if you've read other things, you can hold me to it or hold anyone at your table to it later to discuss what you like. This is really just the tip of the malaria research iceberg at the Burnet. And before I tell you about that research, I'll just give you a bit more background on malaria, because we talk about this complex life cycle, but let's actually nut it out. So, you know that it's in the mosquito and the human. Let's actually see how it goes. So I've put my little parasite in blue. Can you see him over there? There's the little parasite. And if it's in the mosquito and then a mosquito comes along, bites a person, and then the parasite goes into the human, and first it travels to the liver. It spends a short amount of time there. Usually you don't actually know you have malaria at this time. It develops a little, and then it goes into the blood. And here's a nice zoomed in version of what it looks like in the blood. And you can see it actually lives in our red blood cells. So it makes a home in our body, and living inside the red blood cell actually means it's protected from the immune system. So it's using our cells to protect itself. That's how terrible it is. And then you can imagine another mosquito comes along, bites this person that's infected, and they've continued the life cycle. So you can see that all these stages are complex, but firstly, there's a lot of stages happening there. The parasite changes a lot, and there's a lot we have to keep up with, really, to try and understand it and develop a vaccine. Here at the Burnet, we actually focus on the blood stage. Well, a lot of our focus is on the blood stage. There are other projects that don't, but today I will focus on the blood stage, when it's really there in the blood. And the reason for this is that most of the symptoms in humans comes when the parasite is in our blood. Obviously, for the parasite to get into our red blood cells, it has to invade. So here I've got a red blood cell, just like all of us have. And again, in blue, I've got the little parasite, and it's trying to sneak its way in. Once it's in, it grows and makes more and more and more parasites, gets really quite big, and then it bursts out. And then you have so many more parasites to invade so many more red blood cells. And that's how you get more and more and more parasites in your body and cause a lot of disease. Some of those can go and sequester in the brain, in the placenta. So obviously there's so many other repercussions to that. So you can imagine that the rationale to try and prevent that invasion is pretty strong. So something we try and do is make vaccines to block that invasion. If we can make a vaccine that makes antibodies that stops that, we can stop the parasite in its tracks, the immune system can jump in, clean it all up, and we can protect our red blood cells from those diseases. So that's one of the main things we focus on here, trying to inhibit that blood stage. But as you can imagine, we need to actually grow these parasites, because in Melbourne, fortunately, we don't actually have malaria. How are we going to research malaria if it doesn't exist? So what someone did a while ago was collect parasites from the blood of an infected person, and after many years, they figured out how to grow it in a lab, so we can actually keep these parasites in continuous culture, and they're literally over in the next building. We get quite possessive of them, and we call them our parasites, because you spend so much time looking after them and feeding them, at the same time knowing how deadly they are. So we give them food, and importantly, we actually give the parasites blood, because they need to live inside that blood. And we grow them, and when we want to do an experiment on them, the first thing we have to do is make sure that they're going okay. So the way we look at it is we take 2 drops of that parasite culture, and we just put it on a little microscope slide, and we smear it out. Then we dip the slide in a stain called Giemsa dye. The beautiful thing about Giemsa is that it stains DNA. And our red blood cells don't actually have DNA. They've evolved because, they're supposed to carry oxygen, and they said no space for anything we can do without. So most of our cells have DNA, but red blood cells actually don't. So red blood cells will not come up, but the parasites have DNA, so you can see them. So we look at them on a microscope, and this is what you see. That's all your normal red blood cells, pretty happy, no parasites. And there's some sad red blood cells with lots of parasites in them. So if you were diagnosing someone, that's not a good sign. But if you were doing an experiment, you're like, okay, my parasites are ready to be tested now. And then we can do our experiments. So there's so many different questions that still have to be answered. And one of them is, this little parasite over here, what should we target in a vaccine? Because often when you look at a virus, sometimes making a vaccine for viruses has been relatively easier because they don't have as many proteins. This is another version of the parasite, and on here I'm just going to list all the different proteins on that. And I don't expect you to remember any of them. We scientists are pretty terrible for acronyms, as you can tell. But really, there's a lot of targets. And to design a vaccine, one of the questions you have to ask is, what should I put in the vaccine? What should I target on that parasite? And you'd think we would know that easily, but it's still, everyone's still trying to work that out to make the most effective vaccine. So today I'll actually focus on 2 different ways we try and answer that question. Really work out what the best targets are. So one of the ways we do that is study the parasite. We think we know a lot about the parasite, but obviously it knows more than us because it keeps outsmarting us. So we really need to clearly understand which of those are most important for invasion. What's the real Achilles heel of the parasite? Let's go straight to that with the vaccine. One of the ways we've done that is a method that was actually developed at the Burnet. So we use this microscope that doesn't look as exciting as the work it can actually produce. And we take our cultures and we synchronize them, and you can actually incubate these parasites with the right gases and the right blood, and you can actually watch invasion. So this method was developed at the Burnet. This was the first time we could actually see a parasite invading, and now you get to watch that. So there's the parasite, ooh, and it's burst out, and it doesn't know which red blood cell to find, so it's interacting with a few of them. You can see one guy there. The arrows do my job for me. You can see there's so many different processes. There's deformation, that's what the DEF refers to. Then there's invasion happening over there, another invasion over here. And to be honest, we knew that they invade, but we didn't quite realize how many different steps have to happen for this. There's another invasion. So you can see just from that one parasite, look how many more red blood cells are now infected. And you can imagine in the next cycle, 48 hours later, each of those is going to burst out and cause the same problem. So this was actually really, really important work to really understand the detailed sequences of events for invasion. The more we understand about the parasite, the more we understand about the targets, the better it is. And it was really important to disseminate this work, and this came together in a really big publication. And you can see some familiar names there. It was led by our director and many other people at the Burnet. The first and second author both work at the Burnet. The third author, sorry, well, they all work at the Burnet, but the third author was actually doing his PhD on this work, and we used to jokingly call him the parasite whisperer, because I don't know, he made that stuff happen. I don't know how he did it. So really, this work has been crucial to understanding the parasite. So on one hand, we ask questions about the parasite. And on the other hand, we ask questions about the human response to the parasite, which is equally important for a vaccine. So a lot of these questions really rely on international collaborations. So I've highlighted just one of the many here. And we have a really long-term ongoing collaboration with the Papua New Guinea Institute of Medical Research, who are also very interested in malaria research. And some of the questions we ask is, how does immunity develop? If you go to many areas in Papua New Guinea, young children will have malaria. They get malaria a few times, but as they get older, they seem to get less and less malaria. And it appears that children actually develop immunity to this malaria. But obviously it takes, 15 years for that. So we want to compress the whole thing and be able to deliver it in a vaccine. But we still don't even know what is the most important part of that immunity. So one of the ways we study this is to take samples from these children that have developed immunity and also from children that haven't. And we say, what's the difference between these samples? What is making you protected and you not protected? So we take these samples and we test it across as many targets as we can get our hands on. And we do this with the help of this machine that we jokingly call the robot. So I have a picture of a robot, even though it doesn't look like a robot. And whenever people start at the Burnet, they're always very sad when you call it the robot and they go in and see that. But this machine is great because using that, you can study thousands and thousands of samples weekly and really ask a lot of questions about your targets. And one of the key things that came from this study was this forest plot, and I'll walk you through it. Over here, we have a long list of all the different targets. Which one should we put in a vaccine? And here we tested 200 samples of young children from Papua New Guinea. And you can see that these proteins up here had a really strong protective response. And these proteins down here had a really weak protective response. This was the first time that someone actually brought together all these targets and asked that question together. People often work on 2 or 3 targets off on their own, but here we actually showed which were the most important ones so that we can actually help prioritize vaccine development. And that's exactly what we did. We put this work together to help prioritize vaccine development for targets. So, as I said, this is just a snapshot and I can go on forever. Anyone who knows me will know I like to talk. But I think I will end there and just highlight that there's so many exciting things happening, and we really just need to get to our 75% target of having a really effective vaccine. And a lot of that is possible because of all the work at the Burnet, from the scientists to people who work in population health, our collaborators internationally. And hopefully together we can really have an impact on those malaria numbers. Thank you.